Today we are discussing about active transport of glucose. We have discussed in our previous lecture that glucose is mainly transported by facilitated diffusion, which is a passive process, but some of the glucose which remains there, that is actively transported, and we will study how this active transport of glucose takes place. Glucose is actively transported by actively transported by reabsorption cells of intestine and kidney. SGLT, that is sodium ion dependent glucose transporter, is mainly involved in active transport of glucose. If anybody asks you what is the main transporter which is involved in active transport of the glucose, the main transporter we have is a SGLT. SGLT is actually the sodium ion dependent glucose transporter, which mainly is responsible for the transport of glucose. But that transport of glucose is actually an active transport of glucose. If active word comes, means it is ATP dependent, that energy dependent. That means ATP is converted here into ADP plus phosphate. So let's see, we will take one of the cells where the active transport of the glucose takes place. We will take the epithelial intestinal cells, and we will see how the absorption of the glucose takes place in an active transport, how this glucose gets transported. We will see on its lumen side as well as on its blood side. Let's make the diagram and we will understand how the active transport takes place. Say for instance, if I am making intestinal epithelium cells or epithelium cells of intestine, say for this is one of the cell here, and this is our epithelial cell here. If I am making it a different color, say this is one more epithelial cell. It is having a presence of a transporter here. Say this is a transporter here. So this is one more epithelial cell, and we have one more epithelial cell here. So we can say these are the intestinal cells, actually epithelium intestinal cells. These are the epithelial intestinal cells. And these cells, they are attached to each other with the help of a junction. We call that junction as a tight junction. Say this is a junction here I am making. This junction is known as a tight junction, which keeps the cells intact or attached to each other. Now this is the side of the intestines. This is the cells, and this part of the cell is located towards the lumen portion. This is the lumen of the intestine. Okay. Now after that we have cells. Then after we have this region. This is the blood region of the. This is the region of the part of the cell which is towards the blood. So this is lumen part of the intestines, and this is the blood which is external located to it. Now absorption of the nutrients, especially the glucose, takes place from the lumen and it is transported towards the blood. So if anybody asks you absorption, why from the absorption takes place? Absorption is from the lumen, then it moves through the cells, epithelial, intestinal cell, then after it reaches to the blood. So transport is from lumen to blood. So glucose gets transported from lumen to the blood. Now on its apical surface, say if we take this one and this is the apical surface. Apical surface is towards the lumen, and this is our basolateral surface. That means on its basal surface we have a presence of blood. So there is a basal surface here, there is a presence of blood. So on its apical surface we have a presence of S gullet. So what is this here? Is the S gullet. That is a sodium ion dependent glucose transporter. And on its basal side we have a presence of sodium potassium pump. So this is a sodium potassium pump, and we have a presence of gullet. That is a transporter which is mainly responsible for facilitated diffusion. There's a glucose transport we are located here, and we are here location of sodium potassium pump, which also acts as a sodium potassium ATPase. Now you have to keep in mind this is a diagram where we have a presence of various transporters on the various regions. If anybody asks you where from the glucose gets transported, the glucose is transported from lumen to the blood. You have to keep in mind in active transport. But the various transporters, what we call are present on the epithelial intestine lining. We have a gullet. As gullet, there is a sodium ion dependent glucose transporter which is located towards the apical surface, which is towards the lumen. Then after we have a sodium potassium pump, and we have a gullet. Both are the transporters, but they are located towards the basolateral surface, and we can call them located towards the blood. This you have to keep in mind. Now one more important thing which we have to keep in mind. Now if we will take this one, say first if we will take this junction, what is the function of the junction? The junction keeps the cells intact. They are tightly attached. So this surface here or this region becomes impermeable to all substances. That means the substances are not allowed to move through these spaces. Those spaces are sealed up by the tight junction. You have to keep in mind. But some of the organic substances, they move here. Say for instance, if we have a sodium potassium pump, what is the function of this sodium potassium pump? This sodium potassium pump, it takes in 2 potassium ions and takes out 3 sodium ions. You know about it. Now these sodium ions, they are allowed to move because some of the inorganic substances, they are allowed to move through this, what you call this one as a tight junction. They are allowed to move through this tight junction. So sodium and some of the inorganic substances are allowed to move through this space. Not all, but some of the inorganic ions are allowed to move through this space. And if the ions are moving through this space of the tight junction, then we can call this movement as paracellular movement. Paracellular movement. So if anybody asks you, what is the paracellular movement? It's simply the movement through the tight junction. That means the sodium ions, we have a sodium potassium pump here. Sodium potassium pump leads in flux of 2 potassium ions coupled with efflux of 3 sodium ions. And these some inorganic ions are allowed to move through this space of the tight junction. So if we take a sodium, it's allowed to move. And this sodium is showing actually the paracellular movement. You have to keep in mind. So the movement through this gap, through this junction, is known as a paracellular movement. Now these sodiums are allowed to move. Now this S gullet, what happens in the S gullet? That is the sodium ion dependent glucose transporter. Now 2 sodium ions are allowed to move in. This is acting as a syn portal because this S gullet is acting because the ion as well as the glucose, they move in the same direction. And when the 2 sodium ions, they move from higher concentration to lower concentration, that means the concentration here is very high because sodiums are moving towards it. That means the concentration of sodium increases. Now the sodium moves from higher concentration to lower concentration. That it moves from lumen to the cytoplasm of the cell. And it also takes in, because the glucose here is very less, because active transport takes in less glucose. The glucose which remains in the lumen, because most of it is taken up by the facilitated diffusion. Some of the glucose which remains here is taken actively, but some of the glucose, that means very less, that means it is moving against the concentration gradient, that means from lower concentration to higher concentration. That means sodium are allowed to move in, you have to keep in mind, and with it the glucose moves. So we can say S gullet is acting as a symporter, while the substance move in the same direction. That means S gullet here, if we take S gullet is acting as a symporter. Symporter. That means while the two substances move in the same direction, that means they move from lumen to the cytoplasm of the cell. You have to keep in mind, I will capsulate the point. We have the movement, that is a paracellular movement, because sodium potassium pump is leading to the influx of 2 potassium ions coupled with efflux of 3 sodium ions. Now this tight junction species allow some of the inorganic substances to move from blood area to the lumen area. When they are allowed to move, that they move through the space of the tight junction, and we call that movement as a paracellular movement. Now this sodium concentration increases in the lumen. Now this sodium is allowed to move through the S gullet. Now this sodium moves, it moves spatially. At kept in mind, it moves from higher concentration to lower. It releases energy. That energy is utilized by the glucose to move up. That energy is utilized by this transporter taking the glucose from lower concentration to higher concentration. That means here the concentration of glucose is very high, and here the concentration of glucose. So here the concentration is very high, and here the glucose concentration is very low. So it moves from a lower concentration to higher. That is again the concentration gradient. Now that glucose which is taken in here, the concentration increases. You have kept in mind, when the two substances move in the same direction by a same transporter, then we can call that one as a symporter. So gullet, S gullet is acting as a symporter. Then after what happens, this is very interesting point. Now the glucose concentration is very high here. Now we have the presence on its basolateral surface, or in the basal surface, we have a presence of a gullet, that is a glucose transporter only. Now this glucose is taken in. It is taken passively from higher concentration to lower concentration by a facilitated diffusion. Now it is into the bloodstream. Now this glucose is taken into the bloodstream. You have to keep in mind. This is very important thing. So I will recapitulate one more time. It is very important to understand. Here the glucose is showing this type of movement. Tell me, the transcellular movement, because it moves through the cell. It's a transcellular movement. So if we will take this active transport in intestinal cells, what happens actually on the intestinal cells? We have a presence of the transport like S gullet we have towards the lumen onto the apical surface of the intestinal cells. Similarly, onto the basal surface, we have a presence of sodium potassium pump and the gullet, that is a glucose transporter, which is mainly responsible for facilitated diffusion. Now, these cells are intact, they are tightly bonded to each other with the help of a tight junction. Now, the very interesting point is sodium potassium pump leads to the influx of 2 potassium ions coupled with efflux of 3 sodium ions. Now, these sodium ions, they increase in the blood, they move through this space of the tight junction towards the lumen area by a movement known as a paracellular movement. Now the sodium concentration in the lumen, it gets increased. Now 2 sodiums are taken in by this S gullet. When the energy is released, the glucose is also taken in the same direction, but glucose is taken from lower concentration to higher concentration, but sodium are taken from higher concentration to lower concentration. Now the concentration of glucose here increases. Now this glucose is taken up by the gullet, that is a glucose transporter only, that is helping in a facilitated diffusion, that is a passive diffusion from higher concentration to lower concentration. Now this glucose, it reaches into the bloodstream. But the glucose, it moves towards the cell, it enters from one side, then it leaves the cell on another side. We can call this type of movement as a transcellular movement. Now we have a very important thing which we have to keep in mind, that is, if this tight junction becomes dysfunctional, if this is a junction here which is located between the 2 cells, this junction, if this tight junction becomes dysfunctional, say this is a tight junction, if this tight junction becomes dysfunctional, what will happen? Now what will happen? This blood, if this tight junction becomes, say if this tight junction becomes dysfunctional, now this blood will come, it will move through this space and it will reach into the lumen. So the lumen gets filled up by the blood. First thing. Second thing, the microbes which are present in a lumen, they will enter into the blood. That means they will enter into the blood. Microbes will enter into the blood. So blood will ooze out from the extracellular, or we can say the blood will ooze out here, that will come and move into the lumen of the intestine. Similarly, the lumen is filled up by the microbes. These microbes will enter into the blood. If this becomes dysfunctional, but not dysfunctional, in normal conditions, it is what? It's intact, it's compactly packed. So this is how the active transport of glucose takes place. So you have to keep in mind, active transport is here because it's a secondary active transport which takes place here. Primarily we have to see there's an ATP formation, then ATP gets broken down into ATP. So we can say there's only the taking in of the sodium, then initially we have the usage of here ATP. ATP gets used here, you know about it. Because sodium potassium pump is acting as a ATP. Then after that energy which gets, that sodiums which were here present now, these sodium are taken to the lumen, okay, then sodiums will move from higher concentration to lower concentration because energy has been used here, so it is active transport. Sodium potassium pump has used the energy. Then that sodium which was taken into the blood, now this sodium moves through this paracellular movement into the lumen. Now this sodium is used here, okay, that sodium moves from higher concentration to lower concentration into the cytoplasm of the cell and takes with it the glucose from lower concentration to higher concentration. We call it transcellular movement. Then that glucose, it moves through the facilitated entries into the bloodstream. This is actually the active transport of glucose, which is mainly dependent upon sodium glucose transporter, which is acting as a symporter. Tomorrow we'll discuss some more sodium dependent glucose transporters in detail.